the second part and last part of this fifth session. So we will start this last part with a talk from Emilia Moreno, who is coming from the University of Buenos Aires in Argentina. And we will talk about the differences in habituation and dishabituation between pollen and nectar foragers that contribute to foraging division of labor. So floor is yours, Emilia.
Hello, this is Emilia Moreno. I am a PhD student from the Universidad de Buenos Aires in Argentina and a doctoral fellow of CONICET in the Laboratory of Social Insects. And together with Andrés, we studied how differences in habituation and dishabituation between nectar and pollen foragers contribute to foraging division of labor. To begin with, as an introduction, division of labor is a key feature of social insects, which allows colonies to function efficiently. This is Emilia Moreno. I am a PhD student from the Universidad de Buenos Aires in Argentina and a doctoral fellow of CONICET in the Laboratory of Social Insects. And together with Andrés, we studied how differences in habituation and dishabituation between nectar and pollen foragers contribute to foraging division of labor. To begin with, as an introduction, division of labor is a key feature of social insects, which allows colonies to function efficiently by enabling several activities that are simultaneously performed by groups of specialized individuals. In honeybees, workers are sterile females in charge of maintaining a hive. And as they get older, workers go through physiological, hormonal, and behavioral changes that allow them to make a transition from in-hive tasks to perform other tasks outside the nest. For instance, newly emerged bees clean the cells and take care of the brood, while middle-aged bees handle and store food. As the third week of the adult stage, workers initiate foraging outside the nest to collect resources for the colony such as pollen, nectar, water, or resins. In particular, pollen and nectar are the main stimuli that motivate the foraging behavior, and thus the way bees perceive nectar or pollen compounds influences their foraging responses. In this context, while nectar provides energy to the colony, pollen is its main source of proteins. Now, there are fundamental differences in how bees collect and transport nectar and pollen. Once nectar is ingested, it is stored in a portion of their digestive tract called the crop, where it is transported to the hive. Contrary to the way nectar is treated, pollen is not ingested by foragers as it is instead agglutinated and during recollection and transported using specialized structures on the hind legs. Foraging specialization is linked to differences in gustatory perception, particularly in sucrose sensitivity. Previous studies have evaluated sucrose sensitivity of foraging bees by means of the proboscis extension reflex, which is an innate response triggered after touching the bee's antenna with a concentrated sugar solution. When bees were tested at the end of the foraging cycle, pollen foragers were more responsive to a broader spectrum of sucrose concentrations than nectar foragers, which instead responded to highly concentrated sucrose solutions. On the contrary, recent studies show that at the beginning of the foraging trip, pollen foragers were less responsive to sucrose than nectar foragers. Another aspect that is relevant in the foraging context is the ability to learn about environmental cues that predict food sources. Considering that in addition to innate preferences, foraging decisions are also based on learning. When foragers visit a floral patch, they associate the neutral cues of the flower, such as its odors, colors, or shape, with the reward that it provides. After several visits, such associations turn into memories, which influence the orientation of the bees and their landing response, while it can also trigger discrete responses such as the extension of the proboscis. In the laboratory, we can recreate the conditions and parameters in which the bees learn by means of the conditioning of the proboscis extension response. This protocol is based on the paired presentation of a neutral odor to the antennas and sucrose-water solution to the proboscis as an appetitive reward. After several training events, bees will learn that the odor predicts the incoming reward. Although most studies have focused on sucrose responsiveness, a few experiments have also measured gustatory sensitivity to pollen, showing that foragers responded with the pair when they were stimulated using pollen loads. Nevertheless, this response was unstable and bees stopped responding after several events. To deal with this, we developed a protocol in which we rewarded the bee during acquisition trials with a double reinforcement, as we presented the neutral odor to the antenna paired with the presentation of sucrose solution to the proboscis and pollen to the tarsi of the first pair of legs, where honeybees present gustatory sensilla that are hair-like structures in charge of gustatory detection. Using this protocol, we showed that pollen foragers learned odors faster and retained memories better than nectar foragers when we use sucrose paired with pollen as reward, suggesting that both groups of foragers perceive pollen-related cues differently. Nevertheless, we know very little about the possible differences in the responses of foragers when they learn in a non-associative way. In this context, habituation is a type of non-associative learning characterized by a gradual decrease in the probability or intensity of an innate behavioral response due to the repeated and frequent presentation of certain stimuli. This habituated response can be dishabituated by presenting the same stimuli in higher intensities or by presenting a different stimuli but equivalent. Our hypothesis is that worker bees differ in their habituation and dishabituation capacities because they perceive task-related stimuli differently. If pollen foragers are more sensitive to pollen than nectar foragers, we predict that the probability of pair habituation will be lower in pollen foragers than in nectar foragers when we offer pollen plus sucrose together as a stimuli. For this, we trained bees to visit a foraging station and we captured pollen and nectar foragers at the beginning of their foraging trip from feeders with crushed pollen and 10% sucrose water solution. In our experiment, the habituation procedure consisted in the repeated presentation of 25 habituation events. Each event lasted 1 second and was followed by an intertrial interval of 10 seconds. Bees were habituated with either 10% sucrose water solution on the antenna paired with hand-collected kiwi pollen on the first tarsi, or by 10% water solution on the antenna paired with a mechanical stimulation produced by the contact of a clean wooden stick on the first tarsi as control. These habituation trials consisted in the repeated presentation of 4 dishabituation events. That consisted in the presentation of sucrose 50% plus pollen in the case of bees habituated with sucrose 10% plus pollen and sucrose 50%. Bees habituated with 10% sucrose were habituated with either 10% sucrose paired with pollen or 50% sucrose. As results of the habituation trials, to the left you have the habituation curves of pollen and nectar foragers, and to the right the graph shows the pair probability of pollen and nectar foragers to different stimuli during habituation. The x-axis corresponds to the stimuli used to habituate the bees, and the y-axis corresponds to the probability that the pair occurs. Orange lines correspond to nectar foragers and blue lines to pollen foragers. Our results show that when we use 10% sucrose water solution paired with pollen as a stimuli, the probability of pair occurrence in pollen foragers is higher than in nectar foragers. While there are no differences in terms of habituation between foragers under the control treatment. As for the dishabituation trials, the x-axis corresponds to the dishabituation stimuli and the y-axis to the pair probability. To the right, you have bees habituated with 10% sucrose plus pollen, and to the left, bees habituated with 10% sucrose. We can see that pollen foragers habituated with 10% sucrose presented a higher probability of pair as they dishabituate more than nectar foragers when we use 10% sucrose plus pollen as a stimuli. We found no differences between foragers when we use 50% sucrose as dishabituated stimuli, neither when the response was learned with sucrose plus pollen. To conclude, our results support the hypothesis that foragers perceive pollen differently as they perform differently during habituation and dishabituation trials. Pollen foragers become less habituated to pollen stimulus but dishabituate faster than nectar foragers. This difference might contribute to the foraging division of labor. A low habituation and a high dishabituation when pollen is used as a stimuli suggests that pollen foragers are more sensitive to this resource. Thank you for your attention.
Thank you, Emilia. You can come back with us. Thank you for the talk. So that was really nice. While we are waiting for some question on the chat, I have already one question, especially regarding your conclusion. So you say in the conclusion that this difference might contribute to the foraging division of labor. And I was wondering if simply it will not be possible to think that it is a foraging division of labor that is triggering this difference. I mean, I don't know if it's clear what I'm saying, but just the fact that pollen foragers, since they are really, like you say in the sentence just under, very like they are in contact with pollen all the time, they are definitely more sensitive, which obviously will trigger some difference in terms of the habituation and dishabituation performance.
Okay, hi everyone. I'm not sure if I understood well, but the thing is that there are some, or well, I think that there are some flowers that maybe they can produce both rewards, and maybe sometimes bees can be recollecting pollen and or nectar, but they can be with the research. And also inside the hive, they are always both foragers types are always sensing the cues and signals from pollen. So I don't know if I understand what you're saying, that when pollen foragers are collecting pollen, they are more in contact with pollen, so that's why they habituate more.
Yeah.
Yeah, yes, that's exactly, that's what we think too. The thing is, the interesting part is why nectar foragers, what's the difference, why nectar foragers sense the pollen differently. So we think that maybe pollen is creating a kind of an aversive effect on nectar foragers, as they don't like they don't prone to collect it. So yes, we agree with what you're saying.
No,
But
okay. Yeah, I think I was just confused by the sentence because it was sounding like the difference contribute to the foraging division of labor. But in my brain, it will make more sense to say that the foraging division of labor is triggering the difference in the results.
Yeah. Oh, yes. Now I understand what you're saying. Yes. It's the way that we formulate the sentence.
Yeah, so I just wanted to be sure. Okay. Thank you for your answer. So we received some question from AB George on the chat. So like first he's telling that it was a great talk. He's asking, do you think there will be differences in the PR response based on when the foragers are tested? For instance, before flying out and when returning from a food source?
Yes, we think that because now we tested just at when they arrive and when they departure from the feeder. Other studies have tested similar things when they depart from the hive and when they arrive to the hive. Those cues and signals inside the hive, they may, and these contextual cues may influence these differences in the sucrose responsiveness, but also in learning. So that's what we hypothesize, that these differences in the sucrose responsiveness will translate to the learning abilities of bees too. So yes, those cues and those signals from inside the hive, or the cues and signals that they can receive outside the hive during foraging or in the feeder while they are recollecting, yes, they will influence definitely and change these responses. So I think that it's important that we make these, we can ask bees in different foraging moments, when they arrive, when they departure from the feeder, but also when they arrive and when they departure from the hive, but also inside the hive while before they unload pollen loads, or maybe after, because all these cues that they may be sensing may, of course, influence their responses.
Thank you. So we have, we received also a message from David McCluskey who say thank you for a fascinating talk. And we receive another question from maybe George, who's asking why pollen foragers are better at odor acquisition? Has it to do with how important pollen versus nectar is for the colony?
Yes, that's a good question. We have made some experiments between nectar and pollen foragers, testing their olfactory sensitivity and using an electroantennagram, and then we took those differences to behavioral responses with the pair. And so pollen foragers responded more to odors at lower concentrations. So maybe these than nectar foragers. So we found no differences when the concentrations of the others that we tested were high, but when they were low concentrations, we found differences. So I think that pollen foragers may be more sensitive to pollen cues in the detection, in the peripheral detection with at the level of the antennas, and these can translate to behavioral responses. So when we make the pair testing, pollen foragers were more responsive, or they learn better, or they learn better than nectar foragers. And we think that is because they can detect these these others better than nectar foragers at lower concentrations.
Okay, thank you for the answer. I think we are running out of time now for your presentation, but like thanks a lot. And like if anyone has any further question, like you can go on Discord and ask Emilia. Thank you, Emilia.
Thank you. Thank you.